When we consider how a drug will work in the body, we first think about how a drug will look on paper. This is the molecular structure of the drug. It provides a two-dimensional snapshot of how atoms are bonded through space to form a therapeutic. While we often visualize drugs in their two-dimensional form, their 3D reality can reveal an important feature called chirality. If we don't consider chirality, the consequences may be devastating for drug development. When a carbon atom of a drug has 4 different atoms or groups of atoms attached, their possible arrangement in space can vary. If we look at this 3D representation of these possible configurations, the wedge and dashed lines show the position of the orange and green groups relative to the carbon atom. As a result, we have a mirror image. If we look at our hands, we have a left and a right hand orientation of our fingers arranged through space, which are mirror images. They look identical, however, they can't be superimposed. This is chirality. The different configurations are referred to as chiral stereoisomers. The opposite scenario to chirality is achirality. An example of this would be a nail, where the mirror images of the nail are superimposable. Chirality is not something which is limited to drugs. It can also be found throughout nature. For example, our own DNA structure has chirality. The question is why is this significant to drugs? If something looks the same, should it not also have the same therapeutic effect? Thalidomide was a sedative drug prescribed in the 1960s to treat morning sickness and insomnia in pregnant women. If we look at the molecular structure of thalidomide, the chiral center marked by the red circle results in the existence of 2 different configurations. OR and S are terms used to describe the configurations. The OR configuration is an effective sedative. The S configuration, however, led to severe birth defects for thousands of newborn babies. Different spatial arrangements may lead to different types of interactions with the body's receptors. Some configurations, like the S configuration of thalidomide, can have damaging off-target effects, while others, like the OR configuration of ibuprofen, will have an absence of effect. Some may fit the drug target receptors better, leading to an enhanced therapeutic effect. Another type of chirality can exist when restricted rotation around a single bond between carbon atoms in a drug occurs. Bulky groups of atoms or single atoms, indicated by blue, green, orange, and red circles, block the bond's rotation, resulting in different configurations. This is called atropisomerism. As with chiral molecules, varied biological effects of individual configurations or isomers are possible. At the University of Coimbra, we are investigating a light-activated drug called retiquerfin to understand whether an individual configuration can be exploited for an even better anti-cancer therapy. We have to be conscious that even though a drug might look the same on paper, 3D evaluations of a drug's configuration are essential to realize the drug's full potential and avoid unwanted serious side effects.